The CIE 1931 color spaces are the first defined quantitative links between A, physical pure colors in the electromagnetic visible spectrum, and B, physiological perceived colors in human color vision. The mathematical relationships that define these color spaces are essential tools for color management. They allow one to translate different physical responses to visible radiation in color inks, illuminated displays, and recording devices such as digital cameras into a universal human color vision response. CIE 1931 RGB color space and CIE 1931 XYZ color space were created by the International Commission on Illumination in 1931. The CIE XYZ color space was derived from a series of experiments done in the late 1920s by William David Wright and John Guild. Their experimental results were combined into the specification of the CIE RGB color space, from which the CIE XYZ color space was derived. Tristimulus values. The human eye has three kinds of cone cells, which sense light, with spectral sensitivity peaks in short, middle, and long wavelengths. These cone cells underlie human color perception under medium and high brightness conditions. Thus, three parameters, corresponding to levels of stimulus of the three types of cone cells, can in principle describe any color sensation. Weighting a total light power spectrum by the individual spectral sensitivities of the three types of cone cells gives three effective stimulus values. These three values make up a Tristimulus specification of the objective color of the light spectrum. The three parameters, noted S, M, and L, can be indicated using a three-dimension space, called LMS color space, which is one of many color spaces which have been devised to help quantify human color vision. A color space maps a range of physically produced colors to an objective description of color sensations registered in the eye, typically in terms of Tristimulus values, but not usually in the LMS space defined by the cone spectral sensitivities. The Tristimulus values associated with a color space can be conceptualized as amounts of three primary colors in a trichromatic additive color model. In some color spaces, including LMS and XYZ spaces, the primary colors used are not real colors, in the sense that they cannot be generated with any light spectrum. The CIE XYZ color space encompasses all color sensations that an average person can experience. It serves as a standard reference against which many other color spaces are defined. A set of color matching functions, like the spectral sensitivity curves of the LMS space but not restricted to be non-negative sensitivities, associates physically produced light spectra with specific Tristimulus values. Consider two light sources made up of different mixtures of various wavelengths. Such light sources may appear to be the same color. This effect is called metamerism. Such light sources have the same apparent color to an observer when they produce the same Tristimulus values, no matter what are the spectral power distributions of the sources. Most wavelengths will not stimulate only one type of cone cell, because the spectral sensitivity curves of the three types of cone cells overlap. Certain Tristimulus values are thus physically impossible. And LMS Tristimulus values for pure spectral colors would, in any normal trichromatic additive color space, imply negative values for at least one of the three primaries, since the chromaticity would be outside the color triangle defined by the primary colors. To avoid these negative RGB values, and to have one component that describes the perceived brightness, imaginary primary colors and corresponding color matching functions have been formulated. The resulting Tristimulus values are defined by the CIE 1931 color space, in which they are denoted X, Y, and Z. Meaning of X, Y, and Z. When judging the relative luminance of different colors in well-lit situations, humans tend to perceive light within the green parts of the spectrum as brighter than red or blue light of equal power. The luminosity function that describes the perceived brightnesses of different wavelengths is thus roughly analogous to the spectral sensitivity of M cones. The CIE model capitalizes on this fact by defining Y as luminance. Z is quasi-equal to blue stimulation, or the S-cone response, and X is a mix of cone response curves chosen to be non-negative. The XYZ Tristimulus values are thus analogous to, but not equal to, the LMS cone responses of the human eye. Defining Y as luminance has the useful result that for any given Y value, the XZ plane will contain all possible chromaticities at that luminance. CIE standard observer, due to the distribution of cones in the eye, the Tristimulus values depend on the observer's field of view. To eliminate this variable, the CIE defined a color mapping function called the standard observer, to represent an average human's chromatic response within a 2-degree arc inside the FOV. This angle was chosen owing to the belief that the color-sensitive cones resided within a 2-degree arc of the FOV. Thus the CIE 1931 standard observer function is also known as the CIE 1931 2-degree standard observer. A more modern but less used alternative is the CIE 1964 10-degree standard observer, which is derived from the work of Stiles and Birch, and Sporenskaya. For the 10-degree experiments, the observers were instructed to ignore the central 2-degree spot. The 1964 supplementary standard observer function is recommended when dealing with more than about a 4-degree field of view. Both standard observer functions are discretized at 5 nm wavelength intervals from 380 nm to 780 nm and distributed by the CIE. The standard observer is characterized by 3 color matching functions. The derivation of the CIE standard observer from color matching experiments is given below, after the description of the CIE RGB space. Color matching functions. The CIE's color matching functions, under the numerical description of the chromatic response of the observer. They can be thought of as the spectral sensitivity curves of 3 linear light detectors yielding the CIE Tristimulus values X, Y and Z collectively. These 3 functions are known as the CIE standard observer. The Tristimulus values for a color with a spectral power distribution are given in terms of the standard observer by. Where is the wavelength of the equivalent monochromatic light? Other observers, such as for the CIE RGB space or other RGB color spaces, are defined by other sets of 3 color matching functions, and led to Tristimulus values in those other spaces. The values of X, Y, and Z are bounded if the intensity spectrum is bounded. CIE XY chromaticity diagram and the CIE XYY color space. Since the human eye has 3 types of color sensors that respond to different ranges of wavelengths, a full plot of all visible colors is a 3-dimensional figure. However, the concept of color can be divided into 2 parts, brightness and chromaticity. For example, the color white is a bright color, while the color gray is considered to be a less bright version of that same white. In other words, the chromaticity of white and gray are the same while their brightness differs. The CIE XYZ color space was deliberately designed so that the Y parameter was a measure of the brightness or luminance of a color. The chromaticity of a color was then specified by the 2 derived parameters X and Y, 2 of the 3 normalized values which are functions of all 3 Tristimulus values X, Y, and Z. The derived color space specified by X, Y, and Y is known as the CIE XYY color space and is widely used to specify colors in practice. The X and Z Tristimulus values can be calculated back from the chromaticity values X and Y and the Y Tristimulus value. The figure on the right showed the related chromaticity diagram. The outer curved boundary is the spectral locus, with wavelengths shown in nanometers. Note that the chromaticity diagram is a tool to specify how the human eye will experience light with a given spectrum. It cannot specify colors of objects, since the chromaticity observed while looking at an object depends on the light source as well. Mathematically, X and Y are projective coordinates and the colors of the chromaticity diagram occupy a region of the real projective plane. The chromaticity diagram illustrates a number of interesting properties of the CIE XYZ color space. The diagram represents all of the chromaticities visible to the average person. These are shown in color and this region is called the gamut of human vision. The gamut of all visible chromaticities on the CIE plot is the tongue-shaped or horseshoe-shaped figure shown in color. The curved edge of the gamut is called the spectral locus and corresponds to monochromatic light, with wavelengths listed in nanometers. The straight edge on the lower part of the gamut is called the line of purples. These colors, although they are on the border of the gamut, have no counterpart in monochromatic light. Less saturated colors appear in the interior of the figure with white at the center. It is seen that all visible chromaticities correspond to non-negative values of X, Y, and Z. If one chooses any 2 points of color on the chromaticity diagram, then all the colors that lie in a straight line between the 2 points can be formed by mixing these two colors. It follows that the gamut of colors must be convex in shape. All colors that can be formed by mixing 3 sources are found inside the triangle formed by the source points on the chromaticity diagram. An equal mixture of 2 equally bright colors will not generally lie on the midpoint of that line segment. In more general terms, a distance on the XY chromaticity diagram does not correspond to the degree of difference between 2 colors. In the early 1940s, David McAdam studied the nature of visual sensitivity to color differences, and summarized his results in the concept of a McAdam ellipse. Based on the work of McAdam, the CIE 1960, CIE 1964, and CIE 1976 color spaces were developed, with the goal of achieving perceptual uniformity. Although they were a distinct improvement over the CIE 1931 system, they were not completely free of distortion. It can be seen that, given 3 real sources, these sources cannot cover the gamut of human vision. Geometrically stated, there are no 3 points within the gamut that form a triangle that includes the entire gamut. Or more simply, the gamut of human vision is not a triangle. Light with a flat power spectrum in terms of wavelength corresponds to the point equals. Definition of the CIE XYZ color space, CIE RGB color space. The CIE RGB color space is one of many RGB color spaces, distinguished by a particular set of monochromatic primary colors. In the 1920s, W. David Wright and John Guild independently conducted a series of experiments on human sight which laid the foundation for the specification of the CIE XYZ color space. The experiments were conducted by using a circular split screen 2 degrees in diameter, which is the angular size of the human fovea. On one side of the field a test color was projected and on the other side, an observer adjustable color was projected. The adjustable color was a mixture of 3 primary colors, each with fixed chromaticity, but with adjustable brightness. The observer would alter the brightness of each of the 3 primary beams until a match to the test color was observed. Not all test colors could be matched using this technique. When this was the case, a variable amount of one of the primaries could be added to the test color, and a match with the remaining 2 primaries was carried out with the variable color spot. For these cases, the amount of the primary added to the test color was considered to be a negative value. In this way, the entire range of human color perception could be covered. When the test colors were monochromatic, a plot could be made of the amount of each primary used as a function of the wavelength of the test color. These 3 functions are called the color matching functions for that particular experiment. Although Wright and Guild's experiments were carried out using various primaries at various intensities, and although they used a number of different observers, all of their results were summarized by the standardized CIE RGB color matching functions, and obtained using 3 monochromatic primaries at standardized wavelengths of 700 nm, 546.1 nm and 435.8 nm. The color matching functions are the amounts of primaries needed to match the monochromatic test primary. These functions are shown in the plot on the right. Note that and R0 at 435.8, and R0 at 546.1 and R0 at 700 nm, since in these cases the test color is one of the primaries. The primaries with wavelengths 546.1 nm and 435.8 nm were chosen because they are easily reproducible monochromatic lines of a mercury vapor discharge. The 700 nm wavelength, which in 1931 was difficult to reproduce as a monochromatic beam, was chosen because the eye's perception of color is rather unchanging at this wavelength, and therefore small errors in wavelength of this primary would have little effect on the results. The color matching functions and primaries were settled upon by a CIE Special Commission after considerable deliberation. The cutoffs at the short and long wavelength side of the diagram are chosen somewhat arbitrarily. the human eye can actually see light with wavelengths up to about 810 nm, but with a sensitivity that is many thousand times lower than for green light. These color matching functions define what is known as the 1931 CIE Standard Observer. Note that rather than specify the brightness of each primary, the curves are normalized to have constant area beneath them. This area is fixed to a particular value by specifying that. The resulting normalized color matching functions are then scaled in the RGB ratio of 1, 4.5907 0.0601 for source luminance and 72.096 1.37911 for source radiant power to reproduce the true color matching functions. By proposing that the primaries be standardized, the CIE established an international system of objective color notation. Given these scaled color matching functions, the RGB Tristimulus values for a color with a spectral power distribution would then be given by. These are all inner products and can be thought of as a projection of an infinite-dimensional spectrum to a three-dimensional color. Grassmann's law, one might ask, why is it possible that Wright and Gould's results can be summarized using different primaries and different intensities from those actually used? One might also ask, what about the case when the test colors being matched are not monochromatic? The answer to both of these questions lies in the linearity of human color perception. This linearity is expressed in Grassmann's law. The CIE RGB space can be used to define chromaticity in the usual way. The chromaticity coordinates are G where construction of the CIE XYZ color space from the Wright Gould data, having developed an RGB model of human vision using the CIE RGB matching functions, the members of the Special Commission wished to develop another color space that would relate to the CIE RGB color space. It was assumed that Grassmann's law held, and the new space would be related to the CIE RGB space by a linear transformation. The new space would be defined in terms of three new color matching functions, and as described above. The new color space would be chosen to have the following desirable properties. The new color matching functions were to be everywhere greater than or equal to 0. In 1931, computations were done by hand or slide rule, and the specification of positive values was a useful computational simplification. The color matching function would be exactly equal to the photopic luminous efficiency function V, I for the CIE standard photopic observer. The luminance function describes the variation of perceived brightness with wavelength. The fact that the luminance function could be constructed by a linear combination of the RGB color matching functions was not guaranteed by any means but might be expected to be nearly true due to the near-linear nature of human sight. Again, the main reason for this requirement was computational simplification. For the constant energy white point, it was required that x equals y equals z equals 1/3. By virtue of the definition of chromaticity and the requirement of positive values of x and y, it can be seen that the gamut of all colors will lie inside the triangle, 1, 0, 0, 0, 0, 1. It was required that the gamut fill this space practically completely. It was found that the color matching function could be set to 0 above 650 nm while remaining within the bounds of experimental error. For computational simplicity, it was specified that this would be so. In geometrical terms, choosing the new color space amounts to choosing a new triangle in RG chromaticity space. In the figure above right, the RG chromaticity coordinates are shown on the two axes in black, along with the gamut of the 1931 standard observer. Shown in red are the CIE XY chromaticity axes which were determined by the above requirements. The requirement that the XYZ coordinates be non-negative means that the triangle formed by CR, CG, CB must encompass the entire gamut of the standard observer. The line connecting CR and CB is fixed by the requirement that the function be equal to the luminance function. This line is the line of zero luminance, and is called the Illumination. The requirement that the function be 0 above 650 nm means that the line connecting CG and CR must be tangent to the gamut in the region of KR. This defines the location of point CR. the requirement that the equal energy point be defined by x equals y equals 1/3 puts a restriction on the line joining CB and CG, and finally, the requirement that the gamut fill the space puts a second restriction on this line to be very close to the gamut in the green region, which specifies the location of CG and CB. The above described transformation is a linear transformation from the CIE RGB space to XYZ space. The standardized transformation settled upon by the CIE Special Commission was as follows: the numbers in the conversion matrix below are exact, with the number of digits specified in CIE standards. While the above matrix is exactly specified in standards, going the other direction uses an inverse matrix that is not exactly specified, but is approximately. The integrals of the XYZ color matching functions must all be equal by requirement 3 above, and this is set by the integral of the photopic luminous efficiency function by requirement 2 above. The tabulated sensitivity curves have a certain amount of arbitrariness in them. The shapes of the individual X, Y and Z sensitivity curves can be measured with a reasonable accuracy. However, the overall luminosity curve is subjective, since it involves asking a test person whether two light sources have the same brightness, even if they are in completely different colors. Along the same lines, the relative magnitudes of the X, Y, and Z curves are arbitrary. Furthermore, one could define a valid color space with an X sensitivity curve that has twice the amplitude. This new color space would have a different shape. The sensitivity curves in the CIE 1931 and 1964 XYZ color spaces are scaled to have equal areas under the curves. See also, trichromacy, imaginary color, lab color space, standard illuminant, the definition of white point used by CIE and commonly shown in color space diagrams as E, D50 or D65, references. Further reading, Broadbent, Arthur D. A Critical Review of the Development of the CIE 1931 RGB Color Matching Functions. Color Research and Applications 29, 267 €272 doi, 10.1002/col 20020. This article describes the development of the CIE 1931 chromaticity coordinates and color matching functions starting from the initial experimental data of W. D. Wright and J. Guild. Sufficient information is given to allow the reader to reproduce and verify the results obtained at each stage of the calculations and to analyze critically the procedures used. Unfortunately, some of the information required for the coordinate transformations was never published, and the appended tables provide likely versions of that missing data. Chzuna, Pat W. Derivation of the 1964 CIE 10 degree XYZ color matching functions and their applicability in photometry. Color Research and Application 26, 67 €75 doi, 10.1002/1520-6378. 200,102, 26 1 less than 67 AIDCOL 7 greater than 3.0 CO. 2-4, Wright, William David. Golden Jubilee of Color in the CIE. The historical and experimental background to the 1931 CIE system of colorimetry. Inshander, Janos. Colorimetry. Wiley Interscience PPA 9 €24 doi, 10.1002/9,780,470,175,637 ch2. ISBN A978-0-470-04904-4. External links, Introduction to Color Science, William Andrew Steer. EFG's Color Chromaticity Diagrams Lab Report and Delphi Source, CIE Color Space, Gernot Hoffman, Annotated Downloadable Data Tables, Andrew Stockman and Lindsay T. Sharp. Calculation from the original experimental data of the CIE 1931 RGB Standard Observer Spectral Chromaticity Coordinates and Color Matching Functions, colorimetric data useful for calculation, in various file formats, ColorLab MATLAB Toolbox for color science computation and accurate color reproduction. It includes CIE standard Tristimulus colorimetry and transformations to a number of nonlinear color appearance models. Precise color communication Konica Minolta sensing.